A lot of the talks have I have led to some of the slides I'll show you, especially with respect to the impurity control limits for largest unspecified impurity. That's kind of something that's at the back of our mind, and we're working a lot to try to address this. So my talk today, I'm from Polypeptide. I'm based in Torrance in California. And my talk today will talk about how do we tailor our control strategies to meet specific requirements for peptide impurity. Again, the focus on the largest unspecified impurity, which for the analytical scientist is kind of the biggest puzzle. Because we can plan for a lot of things, but during production of a complex peptide, there could be all kinds of small changes or minute changes that could introduce new impurities. So it's a very important and hot topic for us. So I'll talk a little bit about, again, impurity control strategies and how they relate to controlling the incoming starting material, our FMOC amino acid building blocks, and then how are we going to control the manufacturing process. That less focus will be given on here. There's, of course, a lot of science that has evolved in the last 20, 30 years on refining coupling strategies, avoiding racemization. There's just so much beautiful chemistry that was done in the last 20, 30 years. We have a lot of strong scientists here that have contributed to this field. And again, my talk will focus less on this part. And then an interesting part is that we have different customers coming with different requirements and requests. So it's customers and regulators having different visions on what the impurity control strategy should look like, and how low should we go? How low should we go with our largest unspecified impurity? And it's been a very big topic for us. And then finally, just how do we continue to improve this process? Obviously, large manufacturing organizations have commercial products, early stage projects, middle stage projects. So you have a whole dynamic element of a control strategy that's already in place. We're buying starting materials, we're controlling them. We have been for the last 80 years so how do you keep on updating things while the train is rolling, so to speak? So if you break it down, you can look at generating or building a control strategy for a specific peptide really into 5 key elements. The first is, what are you going to do with the starting materials? How do you make sure that you have the right purity and quality attributes for that starting material? And I'll talk a lot about this. You really have to control them on a molar basis, because starting material impurities can have very different extinction coefficients when you measure them by UV. So how does that translate into your peptide? That's going to be very important. The second thing is, of course, as I mentioned, there's been so much work on minimizing impurities to begin with. That being said, impurities could originate from the starting material, or they could form during synthesis. Sometimes it's the exact same impurity that can form by both mechanisms. Sometimes it's different. And then sometimes you have a discount. What's the discount? Well, most peptides today are purified by reverse phase HPLC. So many times you could form an impurity, but live with it because you have a really good way of removing them. So we'll talk about that as well. And then accumulating data. That's actually, in the world of peptides, it's many times very complicated, because by the time a peptide in development reaches commercialization, PPQ batches, sometimes there's only 2 or 3 batches that were produced. So depending on what kind of peptide it is, if you're making a half kilogram batch or a 100-gram batch, that might be sufficient for the whole phase 1 and 2 or maybe 3 program. So many times we are faced with a situation where we don't have a vast array of data or a big data set to understand the chemistry that was collected under GMP conditions. And finally, you can come up with an overall risk assessment which takes into account, first of all, probability of occurrence, and then its impact. So if it occurs, can you remove it or you can't remove it? And then, of course, detectability, which is also a big challenge in the science of peptides. And I would say that a lot of things that came from small molecules have trickled into peptide science. But in the eyes of an analytical chemist, our techniques, they've evolved, and certainly we have now very great UPLC tools, but our separation science is not as capable as it is for small molecules. So there's a lot of challenge, and everyone who's worked with peptides realizes that, sure, you put a dicarboxylic acid or you put a PEG on it and you get these nice wide and broad peaks. So how do you deal with that? So here's kind of the lead into the largest unspecified impurity, which is, again, a big concern for us. So as was just mentioned a moment ago, with the guidance that came out by FDA on peptides that are being synthesized now by solid phase synthesis or synthetic processes that follow the drugs that were approved from DNA origin, example PTH, liraglutide, all of those have largest unspecified impurity limits of less than 0.10%. And if you think about it from a practical perspective, it means that you're forced to live with a process. You have to generate a process that's good enough to be able to not have a surprise in QC, where you suddenly see something that's 0.10 or 0.11, and wow, now you have a new impurity that's never been seen before. And it doesn't sound like a lot, and it is indeed at the edge of what analytical science can give us for peptides. And then we have the other side of the spectrum is the European vision of largest unspecified impurity, which really is kind of hovered around this 0.5% limit, which anything above 0.5%, the expectation is that you would at least know the structure, elucidate the structure, maybe LC-MS or NMR if it's possible. So you'd have an understanding of structural elucidation, and above 1.0%, you'd have an actual qualification. So now you can see the puzzle, because everything between those two limits is possible. And at least our experience at Polypeptide, we've had a lot of different requests from customers coming through that whole spectrum. And indeed, it's not very clear. At the beginning of this conference, Jamie from Bachem gave a talk about some regulatory interactions with FDA on one of their peptides. And it's not very clear there's no clear guidance what should be the limit, how low do you need to go. So again, different sponsors, different regulators have different requests. So in parallel to this evolving regulatory environment, we've embarked on a journey a couple of years ago to update and modernized our control strategy for the starting materials. And this requires method development. And again, I said a lot of the challenge is that you have an ongoing control strategy. You have registered commercial products. How do you implement new products while you have an ongoing supply chain? So method development, understanding impurities, having methods that are much more capable, of course, with UPLC being so readily available and more reliable these days. Going from HPLC to UPLC certainly is a huge leap forward. Much better mass spec capabilities today, I think we had than 10 or 15 years ago. But it's deeper than that, so it's a better understanding of the justification of specifications based on the routes of synthesis. And I don't think it should surprise you that if you go out and buy an amino acid, say, I don't know, FMOC alanine, and you look at 3 different suppliers, they'll make it differently. So the processes are not always identical. In fact, they're most often different. So you really have to understand what the impurities that can form from each process and how are you going to control that. So part of that is the justification of specification, understanding route of synthesis, and finally, method validation, and of course, working with all your customers and the suppliers of the raw material. So this kind of sets the tone for this puzzle that I'm showing you, is how do you do this again with all the activities that you have in a company that's producing commercial products. So just to touch on 2 examples, we in the past 2 years have been working to modernize all our methodologies for the FMOC amino acids, and that included being able to go down to a limit of quantitation in the area of 0.03 to 0.05 mole percent. So everything again is quantified based on relative response factors, so we understand the molar contribution to the final API. Some examples, some amino acids have side chain protecting groups that are very strong absorbing, and that'll be a huge difference. We've had relative response factors ranging all the way down to 0.2, so it's quite significant. If you only control it based on your relative response by UV, you would really be setting yourself up for failure. So we embarked on this journey. We've essentially validated all our methods, and this was interesting. We worked with a couple of customers, and they've asked, although it's not in the guideline, they've asked for a full ICH validation. So if you read ICH Q3 revision 2, the premise of validation is for API and drug product. And for many years, starting materials had pretty limited validation packages. But we've had a lot of customers come to us and say, look, we want the full ICH, robustness, intermediate precision, the whole thing. So we've been validating methods to comply as if they were APIs. So it's quite a lot of work. Now, the positive side is you have a very reliable method that you know that you can transfer, and you know that it will perform well and that you'll be able to reproduce it. So this was part of the journey. And then, of course, all these methods, ideally they're mass spec compatible. In our case, we ensured that everything is mass spec compatible. It makes it easier for a QC laboratory as well. So if you're thinking about running a method like this is an example of aspartic acid. The QC lab, if it's mass spec compatible, doesn't have to inject every time they run this method all the standards, right? You can just use mass spec to identify things. So it's really, really helpful. So in parallel to this, we also needed to update our control of the enantiomer methods. And the interesting puzzle about the enantiomer methods is, yes, we had to go down to very low LOQs, but we also had to ensure that the impurities that are detectable and that are present in the main method, the main HPLC method, don't by chance coalute with the de-enantiomer that we're looking for, or else it's going to be a nightmare in QC. So this was part of the work of development, really teasing out the 5 to 10 impurities that we had for, say, each amino acid, key impurities, and just making sure that that this de-enantiomer is highly specific and that we don't have interference. So here's an example of where we ended up with a control strategy for aspartic acid, kind of the basic control strategy. Of course, there could be cases where you need to go tighter, right? Where you have no choice, you have something that's amplified. I mean, nobody promises you that if you have an impurity, for example, FMOC beta-alanine, which is present probably in every single amino acid, nobody promises you that it'll couple exactly at the stoichiometric ratio that you have here. So You might have good intentions and you're controlling it down to 0.09% mole percent, but it might amplify in the final API. You have to be cautious a little bit. But you can see how we look at these impurities. Some of them can be controlled to a higher level. For example, the FMOC asp OT-butyl, which of course is capped and would not react, right? So some of the impurities you can control you can give yourself a lower level. And as I spoke at the start, maybe one of these impurities is rejected by the downstream purification process anyhow. But that becomes very challenging for a company to maintain. You don't want to have a spec for an amino acid for every single product. That becomes a nightmare. So you want to have some type of basic unified specification for a starting material that's applicable to all peptides. And then as part of our journey, we also replaced TLC. So we still have it here in this version of the spec, but that's kind of going out the door. We're using charged aerosol detecting. HPLC, it's much simpler. TLC has advantages, especially in the discovery phase, but for a QC laboratory, it's really hard to maintain consistently. So we've kind of been on the journey to replace TLC. And another interesting part that we have implemented is really looking at residual acetic acid. And there's, of course, other acids also that could be used in production of other amino acids, but acetic acid is a very kind of common byproduct. And it can actually get you into a lot of trouble. So, of course, it reacts quickly. And so you get capping, and many times you can take a really large yield loss. So if you have, for example, 1,000 ppm, which is not a lot of acetic acid, you can lose 5–10% of your peptide just by that capping. So another important part of the control strategy. So with all this said and in mind, we again embarked on this journey to update and really implement a modernized control strategy, and understanding that this is just the first layer of control. So the design of the process, the use of reagents to minimize racemization, the control of key coupling steps. All of that has to be part of process development in a world where we're developing peptides with a very limited batch, kind of batch history. Sometimes in small molecule chemistry, there's, especially for the oral small molecules, sometimes there could be 50 or 100 batches by the time they get to commercialization. And I think in our world, if we get 5 batches, I consider us lucky. So, how do you generate that knowledge with a pretty limited data set? And then, of course, a big question. Okay, we have been asked to go down to 0.10%. We are able to generate methods to control the amino acids. Can these amino acids be produced this pure? It's not a trivial question. And then cost comes into play. So can you actually bring all of these impurities in the amino acids to such low levels? So that's a big part of the discussions with suppliers that we have. So we talked a little bit about manufacturing process controls. Talked a little bit about difficult couplings and side reactions. Those, again, need to be identified. Typically, a lot of the chemistry, we try to do it at small scale in process development. And by the time we get to GMP, hopefully a lot of that's resolved. But many times, there are surprises. And as you go in and as you scale up, you encounter new impurities that you need to address. But you're collecting knowledge, and as you understand the impurities and whether you can reject them, that really plays into your overall control strategies. Impurities in the crude, I think we're seeing more and more a systematic approach of generating key impurities, whether they're in the crude or forcing the crude into I'd say, forcing the reaction to not fail, but almost fail, just to understand if we have process removal. And then finally, looking to see what the API batches we have when we make GMP batches, which of the impurities should end up in the final control strategy. So interesting debate. We know that for our generic peptides, we need to be better than 0.10%. So the limit is 0.09, right? That's kind of the acceptable limit. In the final API release method. And we already have commercial products out there. There's a lot of pharmacopeal accepted commercial products out there that have a level of 0.5%. So then we have a gamut of different requests coming from customers and from regulators. And it poses a very different burden and challenge on the starting materials. So that interplay is very, very important. As I mentioned, amino acids are made by different processes. Not all the suppliers can meet such low impurity standards. If you ask a supplier that's making FMOC isoleucine to reduce the level of FMOC leucine in that isoleucine, it becomes really, really challenging. So how low can you go? And then again, to add on to this picture, we are making peptides at larger scale than we've ever done before. So peptide volumes are increasing, multi-ton scale now. So multi-ton scale amino acids. A lot of suppliers are making amino acids in 300 or 500 kilogram batches. There's some suppliers that are now moving towards metric ton batches. So single batch of FMOC amino acid would be purchased at a 1 metric ton scale. So obviously a lot of this just gets more complicated and more expensive. And then hopefully within a year or two or three, we'll get better guidance from regulators what the expectation is and what the value proposition also to patient safety and patient health is. If you look at a UPLC method today for any complex peptide, you can see that going down to 0.10% is really at the edge of what we can do from the science perspective. We can get again to LOQs in the area of 0.03 or 0.05, but going down much lower than that is really, really challenging. But 5 or 10 years from now, there'll be newer technologies and even separations that have better resolution. So will the expectation be to go down to even lower levels, and to what effect, right? To what are we actually serving the patient's needs by going down to these extremely low levels, right? So that's kind of also a topic of debate. So hopefully there will be more clear guidance and more clear rationale or decision tree when you would choose 0.5 or when would you choose 0.2 or what's the rationale, what's the indication, and what's the medical need that's being addressed. So kind of in summary, as we're collecting more data, we're having a control strategy that's evolving. Currently we're in commercial supply of a lot of products. So we have methods that are, I call our older methods that are probably implemented between 10 and 20 years ago. And now we're coming up with a new set of methods that's being implemented. So how do we get that into the supply chain and start the journey also of updating regulators, especially for our registered commercial products. So it's a very, from an analytical perspective, also from a procurement perspective, it's a very interesting challenge. how do you purchase and control starting materials, FMOC amino acids, to a very high purity limit with very low levels of impurities? And how do you implement this as part of your life cycle on newer drugs that are coming into the market and also approved products? So with that, my last slide is just to acknowledge some of my colleagues and collaborators at Polypeptide. Anna, who's our AD manager in Malmo, and Vincent and Didier. They've been working with us for many years from our sites in Strasbourg and in Braine. Jan, who's our global head of QA, and he's based out of Malmo, and the Polypeptide team. And thank you very much.